Today is Tuesday, September 16th, 2008. My name is Mark Depew. I'm the Director of Oral History at the Abraham Lincoln Presidential Library. And I'm here with Linda Kull. This is part of a series of interviews we're doing with people at the National Soybean Research Center. Linda, welcome.
Thank you, Mark.
Why don't you start with telling us when and where you were born?
I was born in Illinois, if not left, born in Mount Vernon, Illinois. And around fourth grade, moved all the way up to central Illinois.
Now, maybe you don't want to tell us the birth date, but you do need to say that.
Okay, I was born in 1954.
Okay. Mm-hmm. What day?
May 3rd, 1954.
Okay, and a little bit about growing up then.
Oh, growing up in a small town, I think is wonderful. And it became more wonderful to me as events like 9/11 happened. I felt very comfortable and safe living in central Illinois in a small town. Enjoyed small neighborhoods, being able to run outside and play. The school I went to was just a block down the alley, and that was always convenient.
What did your father do for a living?
My father was an architect. He designed churches and schools. He tried to stay away from homes. But I can drive around the country and almost just central Illinois and recognize the schools and churches that his office designed. They have a similar kind of a Frank Lloyd Wright look about them.
Something of a labor of love, especially doing churches, I would think.
Yes, he usually came home with a headache working with committees and that. It's difficult. You know, when you send something out to committee, you get a lot of different opinions. So school boards and church boards were always a challenge, yes. Yeah,
they're demanding folks, I would think. They have it in their own minds exactly how it should look, huh? Mm-hmm. Mm-hmm. What was your father's name?
Leland Dwight Gatewood.
Okay. High school, did you start developing some interest? Did you know what you wanted to do with the rest of your life in high school?
Yeah, I probably didn't know what I wanted to do with the rest of my life. But as I look back, the interests that I had in high school were indicative of what with what I ended up as a profession. So in high school, I loved science and math and the biological sciences, and I was secretary of the science club. Plants, oh my goodness, enjoyed the study of plants. And that, along with my father being an organic gardener, kind of fed an area of interest that soon became my profession.
Did you have a big garden plot at home?
As big as the backyard would allow, yes. Dad chucked that backyard full of vegetables. There were years that my sister and I found that we were out at the fairgrounds bagging up manure to bring home to the garden. When the community had a leaf drive in the 70s, Dad had hundreds of bags of leaves dumped in the driveway. And we mulched them up, rebagged them, and put them on the garden.
So your chores growing up were a lot to do with that garden, I would think.
In the summertime, they definitely centered around the garden. So in high school, my sister and I had a wonderful exposure to freezing and canning. I learned how to use a pressure canner without blowing up the kitchen.
Now, not every child, every kid takes to things like canning and working in the garden and collecting manure and things like that, but did you enjoy that?
I probably didn't enjoy the manure part. But again, one of the wonderful things of a small town is you have this backyard and you can grow a vegetable garden. And spend weekends and summers with the family doing simple things like canning and freezing. We were a very simple family. We didn't go out much. We didn't eat out much. Mom cooked 3 meals a day. So growing up with those types of events, it was a very simple life.
What did you want to do beyond high school?
When I graduated from high school, I wasn't sure what I wanted to do beyond high school. It was assumed that I would get married and have a family, and didn't realize that somewhere in my future was a PhD in the industry of the soybean industry. It just sort of happened.
Did you go directly to college from high school?
I did. I went to the local junior college and enjoyed that, but I was in the wrong area. I was in the area of business, so learning shorthand and typing and bookkeeping, and quickly found out that that's not what I wanted to do. So I was married and had 3 children, and when the 3rd child was in kindergarten, decided I wanted to go back to school. Mm-hmm. And I went back in the biological sciences, and shortly found that I had a passion, a deep passion for the biological sciences.
What was your husband's profession?
He was schooled in landscape architecture, but his family had a rather large farm in central Illinois, and as his father wanted to retire, then he wanted one of his boys to get into farming. So we came back to the farm in 1976, the bicentennial of the local community, and took over the farm, helped farm, which was about 1,000 acres.
That's a good-sized farm, especially in 1976.
Yes, it was. Yes, it
was. Did you find it to be quite an adjustment from small-town girl to farm girl now?
Oh, it was quite an adjustment. It was quite an adjustment. But what was wonderful about that is Grandma and Grandpa were just down the road, a quarter of a mile down the road, and they had farmed all their lives. So my husband's family had been in farming all their lives, and she was a wonderful resource and help with trying to get me used to living on the farm.
What year did you go back to school?
Back to school in about 1985.
Okay. And I assume you still had some small kids back in the farm then?
So yes, moved to the farm in 76, and the first child was born in 1977, second child born in 1980, and the third child born in 1982. Went back to school when my daughter, the third child, was in nursery school.
And that had to be quite a change too.
It was. But what we did was we had a little minivan, and going to activities, ball games, t-ball games, soccer games, dance lessons, just took the books with me. So we had a minivan and a stack of Mom's books, and we traveled.
And at the games, Mom's nose is in the books anytime there's a break?
Yes, they'd see me there, but I was probably studying as we were watching the games.
What were you majoring on then?
My major was in plant sciences. My first degree was in the botanical sciences, or a botany degree, at Eastern Illinois University.
Okay. And after you got your undergraduate degree at Eastern, what year was that?
Okay, that was in 88, I believe, 1988. Mm-hmm.
And what did you want to do with this degree?
I graduated with a degree in botanical or biological sciences, and also certified to teach in the state of Illinois. So I had a teaching certificate for high school, 9 through 12. Student taught, and decided that I might want to stay in school and work on a master's degree.
Did you do that?
Yes, I did.
At Eastern?
Yes, so a master's degree in botany again, specializing in plant physiology. Finished that in 2 years and graduated in 1990. And stayed at Eastern and taught for 4 years and enjoyed teaching. I was on the other side of the desk.
So you're commuting back and forth during the time you were teaching and obviously going to school?
Yes, but that wasn't a long commute. So
Okay.
that was about 10 miles between university and home.
Do your children enjoy growing up on the farm?
Oh yes, they enjoyed growing up on the farm. Farm kids get to do some things that the city kids don't get to do. We had 3-wheelers and 4-wheelers, and lawnmower tractors that could just whirl around the house at speeds way too fast. We had animals, enjoyed animals, dogs and cats, and an assortment of other critters outside. The kids would bring home snakes and stray cats. We got to shoot BB guns. They enjoyed that. You couldn't do that in town.
Well, that would be a big attraction for me.
We also had chickens at Grandma's house just down the road. So they gathered chickens. There were hogs down there too. So they enjoyed some simple things, some very simple life on the farm.
You're teaching what course at the university?
At Eastern Illinois University, I taught economic botany. That was a fun class to teach. Biology, and a microbiology class.
What happens next?
After 4 years of teaching at Eastern, I realized I really enjoyed the college atmosphere. And in order to stay in the college atmosphere, I needed a PhD. So I decided to, by encouragement from my family, because when you have 3 children and a husband, getting a PhD is a family effort. It takes their support. So I went back to, I actually applied at the University of Illinois in 2 departments, and in one department, which was the Plant Pathology department, one of the USDA and faculty researchers received a grant from a soybean checkoff board, and they needed a PhD student and a master's student, and it was to work on one specific pathogen of soybean. That's Sclerotinia stem rot, still my favorite fungal disease of soybean. And that provided, it was a wonderful opportunity, it provided that PhD student and master's student with a tuition waiver and a stipend.
Okay, there's a couple terms in there that maybe not all of us understand. Soybean pathogen.
So you want to know what a soybean pathogen is?
Well, that would be nice, yeah. I think I know, but just to make sure we establish that as a baseline.
Okay. So soybeans and plants, but we'll focus on soybeans, have pathogens just like people and animals. So soybean pathogens can be fungal or bacterial or viral or worm-like organisms called nematodes. So all of those are major soybean pathogens in Illinois.
And the other term was the Checkoff program. I'm not sure exactly how you phrased it, but the word checkup came up.
Okay.
Checkoff came up.
The checkoff in the agricultural industry refers to farmer funded programs. So checkoff monies are accumulated by an act of Congress provided for, I call it a tax on every bushel or unit, so in soybeans that would be bushel, that's sold for the first time at the point of sale. For soybeans, the tax is 1/2 of 1% of the selling price goes into a checkoff fund.
And the Checkoff fund is used for what then?
The checkoff fund can be used to promote and fund research for that particular commodity.
To fund institutions like this one, perhaps?
Not really institutions, but fund good research at these institutions.
The money is normally distributed through grants then?
Yes, yes. So in Illinois, at the point of first sale, checkoff monies are generated, and that goes to the Illinois Soybean Association, which is a checkoff association. Then a portion of those monies go to the National Checkoff, which is the United Soybean Board. And each one of those boards oversee the checkoff funds and fund research, a lot of research at universities.
Okay. And I think we're getting close now to when you actually came to this institution. So we've got you at the University of Illinois. You're a PhD candidate. Can you connect the dots to this institution?
To the National Soybean Research Laboratory?
Yes.
Okay. So I worked in I did my PhD work in a USDA laboratory that's downstairs here in the National Soybean Research Center. The USDA office, or the USDA lab, is within this building, and this building is called the National Soybean Research Center. The laboratory that I work in is the National Soybean Research Lab, and it's upstairs from the USDA lab, which is just below us here.
So it was a natural progression from you completed your PhD, and then did you apply for positions here?
Mm-hmm. Mm-hmm.
Okay.
So it took about 6 years to complete my PhD, because during that 6 years I also worked as a research specialist with a 100% appointment. That would allow me to begin to work for the university and be included in a benefits package, which is a desirable thing. And then finishing my PhD, I became aware that the National Soybean Research Laboratory was looking for someone to oversee their production research. And the production research was heavily funded by the Illinois Soybean Checkoff, also by federal and state sources.
Tell us what production research is.
With production research, try to grow soybeans with less disease and increase the yield. So production is, how do we produce soybeans? How do you grow more of them with less disease?
Okay. Here's a big shift for us now, but I think you're ready for this one. Can you tell us a little bit, just a very thumbnail sketch of history of soybeans in the United States? And I'm especially intrigued, if you were to look 100 years ago, farmers in Illinois weren't growing soybeans. So how did we come to having it be such a dominant crop?
Well, if you'd want intense information, there's several books available that you could read. And I got my hands on one recently, expecting some questions about the history of soybean in the United States. And I became very intrigued. I could hardly put the book down. I didn't realize that so much had happened in Illinois that contributed to soybeans being a very important economic crop. So in the United States, I'll pull a few dates here. The first time that soybean is known to have been grown or planted in the United States was 1765, and that was in the British colony, not the United States at the time, but the British colony of Georgia. Then around the 1850s is when soybean was grown in the Midwest for the first time, first grown in Ohio, then Illinois and Iowa, and then a few years later in San Francisco. And it was grown mainly for hay or forage for animals, and then as a restorative properties for the soils.
So to put nitrogen back in the soil, for example.
Mm-hmm.
Did they really understand that aspect of it back 60, 80 years ago?
Not fully. That research was yet to be done. Is why soybeans was able to restore certain components that are necessary for other crops in Illinois, like corn, to grow well.
Okay. When was the big explosion when we suddenly saw a dramatic increase in soybean production in the United States?
There were several factors that contributed to the dramatic increase of soybeans in the United States, and that began, say, around the turn of the century, around 1900, when people began to conduct research on soybeans. The first research plot planted at the University of Illinois was in 1896, and they looked at the components of soybean that made it a good silage or a good animal feed. And they also looked at how soybeans could rotate well in crop systems. And also the first soybean variety trials were begun around the turn of the century at the University of Illinois. With research that was at the University of Illinois with some personalities that pioneered soybean genetics and genomics and breeding and agronomics, and that was Burleson, Professors Burleson and Hackelman and Woodworth, with the research that they did, and then also the concurrent research that was going on by George Washington Carver at the Tuskegee Institute in Alabama, in which he looked at the components of soybean, protein and oil, and created and developed over 300 uses for soybean. that looking at soybean as a viable crop for animal feed with the protein and oil composition and the potential for products for human use provided the underpinning for the expansion of the explosion actually of soybean as a cash crop or an economically valuable crop.
So before the early years of the 20th century, then one of the problems with soybeans was nobody knew what to do with them other than as cattle feed, perhaps?
I don't know if it was that. It was more or less we didn't know the value that was there.
And we're going to learn quite a bit more about the value and all the processing steps, and I'm sure that'll be interesting. What are the base components, the value of soybeans that we know of today?
Soybeans are known today for their protein and oil. A soybean seed is about, and it ranges because the environment will influence the oil composition and the protein composition. But soybeans are about 20% oil and about 38 to 40% protein.
Is there any other crop, food crop, that has that high level of protein in it that you know of?
I think soybeans are known for their protein composition.
Yeah, that seems to be a very high content. One of the words you mentioned, genomics. Am I pronouncing that right?
Genomics.
Genomics. Can you describe what that is?
Genomics is the study of the soybean genome and the study of the traits and what they do either functionally or structurally for the plant.
Okay. And let's go into your specific area, the pathology side of it. What are the things that especially threaten soybeans?
Okay, so the things that, say, threaten soybean yield.
Yeah.
In Illinois and in all major soybean production states, there's probably around a dozen pathogens that are continually beating back the yield of soybean.
Are there some in particular? And you mentioned cyst nematode.
Soybean cyst nematode is the number one yield reducing pathogen in Illinois and the world.
And what's the best way that we found to combat that?
Typically for any disease, the best way to combat or control the disease or manage the disease is with resistant varieties. So that would be a farmer's choice, is to plant resistant varieties that aren't affected by the disease.
Well, how much does this institution have to do with developing those resistant varieties?
It's a major focus. In fact, with most soybean research programs or production programs, there's two major focus areas. One would be looking at traits or genes that increase yield, identifying them, and moving them into the varieties that are preferred by growers, and then screening varieties for resistance, and turning those varieties, we're a public institution, making that germplasm or those varieties available for companies so they can incorporate those traits within their germplasm or varieties that are preferred by soybean growers.
So genetic modifications is a very big part of that process and what you do then as well?
Genetic modification would be a little bit different than conventional breeding.
Okay.
So with say germplasm and breeding efforts, that would be done by traditional breeding methods. That would mean you'd produce say a non-genetically modified germplasm or variety.
Germplasm meaning?
Germplasm would be a general term referred to that refers to maybe a variety or a line or a population that's not a named commercially available variety.
Okay. I guess the word that scares people, though, is genetic modification. That's developed a certain reputation, and some people are concerned about that. Can you talk about what it is, and then maybe a little bit about the ethics of it?
Okay. Genetic modification that would tend to concern people would be that process that brings genes or DNA from an organism outside of the soybean genome into the soybean genome. So bringing DNA or genes that are not within the soybean genome could be termed genetic modification.
Do you have any examples of that?
Yes, they're good examples, though.
Okay.
So some of the technologies, we think of technology in agriculture as that piece of equipment like GPS technology sitting on a tractor, but there's other technologies that are just as important. And one of those would be a genetically modified soybean, say like the herbicide-resistant soybean. We know that as, say, a Roundup-ready soybean or a Liberty-Link soybean. So that's a soybean that has a trait or a gene that's not found in the soybean gene pool that gives the soybean a trait that's economically desirable. For example, we can spray herbicides on soybeans, kill the weeds, and the soybeans still stand up.
What would you say to people that would argue that, well, that's just wrong, it's dangerous to introduce that to the food supply?
I would probably say we're all more than welcome to have our opinion about the food we eat and the crops we grow. But what we've found with genetically modified crops and soybean in particular is that those genes are really not harmful to us. And they're advantageous to the environment. That means we may be better stewards of the environment. We spray less of a chemical in the environment. They can lend themselves well and support soil conservation. So when you weigh the positives and negatives, chances are with the genetically modified technologies that we see now, there's more advantages than disadvantages.
Mm-hmm. Soybean rust is another thing that we've heard about increasingly over the years. Mm-hmm. Can you talk about why that's something that's come to the public's attention here recently?
Well, until 2004, soybean rust did not occur in the United States. In the countries in which it did occur, it was a very devastating disease. Very devastating disease. So you can imagine for the ag industry, the soybean industry in the United States to contemplate soybean rust arriving, it was a very awesome thing to think about it. And one of the main reasons that it was a fear is that there are no varieties that we were growing here at that time that had resistance to soybean rust. Plus, as a pathologist, you have an appreciation for what pathogens do and how they do it. There's some pathogens that move in slow, and their life cycle is one that allows the disease to maybe develop slowly. Whereas with soybean rust and other diseases of any economically important crop, there's rusts for corn and rusts for wheat. A rust is a rapid cycling disease, which means it can go through a life cycle or through a cycling event and produce lots of infective, we'll call them propagules, called spores, that can infect, and millions of them, can infect and reinfect in a 1 or 2 week cycle. That's a scary disease.
Does it stay in the soil then after the crop has been harvested?
Rust doesn't overwinter here. Rust is a pathogen that is an obligate pathogen that has to carry on its life cycle in living tissue. It's kind of like a virus. A virus can't live out by itself. It needs a human cell to carry on and recycle itself or renew itself. So with rust needs living green tissue in order to continue to produce more rust spores. So in Illinois, in the winter, when all the plants are dead or they're perennials and their living parts are still underground, the only place in the United States that rust can live through the winter would be in the southern tier states.
Which traditionally haven't been the major producing areas for soybeans in the first place?
Yes, that's true. That's true. So the major soybean production states are not the states in which this pathogen can overwinter.
But it sounds like you've just described a scenario where it's a huge problem in Brazil.
Yes, it is. It is a huge problem in Brazil. When you think of the devastation of the pathogen, there's several things that you need to consider. Actually, 3 things. We'll call it the disease triangle. And it is a triangle. You have the host, and the pathogen, and the environment. And areas in which diseases like rust are very big problems, they're economically important problems, is an environment where you have the host and the pathogen, and is an environment, is the point, where disease can occur in which the host and the pathogen are always there. So in South America, the host and the pathogen are there, and it also has an environment almost year-round that can allow rust to continue to infect.
Now, you alluded to something when we first started this conversation, that at the time when the word came out that soybean rust was starting to migrate into this part of the country, there weren't varieties of soybeans that were resistant. Does that mean that there are soybean varieties now that are resistant?
We're working on that, and that's one of our efforts, and that's one of the efforts that the Soybean Checkoff in most of the soybean production states would be contributing funding to universities to look for rust-resistant varieties.
Are you partnering with any other parts of the world in that process, since it's such a bigger problem in places like Brazil, and I would imagine in places in Asia as well?
Yes, the University of Illinois and most universities are, that's one of their major efforts, is partnering with groups internationally and exchanging students and exchanging information and exchanging soybean germplasm for us with different countries.
Okay. Well, we've covered a lot of terrain here. What have I missed so far that you needed to talk about in terms of the work that you do here?
Oh my goodness.
I know that in the next step we're going to see some things. Go ahead.
Well, since I'm a pathologist, one of the major ongoing challenges is to find resistance genes. And soybean has a very narrow genetic base, which means that all the hundreds of varieties that are grown by soybean producers in the United States come from only a few ancestral varieties. And that can be a problem. Because if you don't have resistance, say to soybean rust or soybean cyst nematode or any economically important pathogen, where do you go find those genes for resistance? Where do you go look for new or novel traits for yield or disease? So you ask somebody that, and it's just, I don't know, where do you go look for novel genes? And there's places. For example, the home of soybean, where soybean originally grew, is China. And also in Australia, there's some exotic relatives of soybean. So for soybean, we can go back when we have researchers and scientists that have gone back to China and the Asian countries, and they look for soybeans that are distant relatives, or say close relatives or distant relatives to the glycine max soybean that we grow today. Or go to Australia and find distant relatives, exotic relatives, say the glycine tomentella. So that's a different species. So in China we find glycine soja, and in Australia we can find glycine tomentella. And in tomentella in Australia, it's growing in ditches as a viney little weed. It's not a very impressive looking plant. We can bring these germplasm lines, these germplasms, back to the United States and begin to screen them for diseases. So these would be traits that we're looking for that aren't in the germplasm or soybean genome that we have access to today. If we identify soybean rust resistance, or high yield traits, then in these plant introductions that we bring back to the United States, then the challenge is how do we get them into our current or elite germplasm that's preferred by soybean growers here in the United States. And that can be a challenge. For glycine soja, they cross very well. So you can, a breeder or an agronomist can make those crosses, moving pollen from one to the flower of the other, and screen those generations, those subsequent generations. For glycine tomentella, that's a bit more of a challenge. And that makes sense because some of the most desirable traits that we found, new traits, we have found in tomentella. That would be resistance to certain diseases that we don't have notable resistance to right now.
What's the timeline, though, to introduce that into the variety of soybeans that's going to be commercially viable?
Okay, and that's a fantastic question, and we answer that question with farmers that we meet here in the lab quite frequently.
Yeah, that's an immediate concern to theirs, I suspect.
Well, we get to sit down and talk to farmers on a regular basis here in the lab, and we ask them, what do you need? And they tell us in the area of weed control and disease control and yield and agronomic practices and disease management practices, what their challenges are. And what we're looking at is, from our point of view, there's short-term deliverables and there's long-term deliverables. In weed science and some agronomic practices, we can start a research program and in a year have a deliverable that can go right to the farmer, and it can be information that he can use or she can use to make decisions. And that's wonderful and that's easy to understand. But to answer your question, there are also long-term deliverables, and usually a breeding for resistance that starts in the public domain or at universities, breeding for high yield traits, those are long-term deliverables. So our investigators start with screening germplasm. The pathologists will screen the germplasm. They'll hand something off to the breeders. The breeders would necessarily start moving those traits and developing markers, genetic markers for those traits, so that as those populations of plants are screened for those specific traits, then we can use markers instead of growing it out in the field once a season and then screening it. Markers are a very quick genetic tool that can be used to screen populations very young, identify those traits, and then move that germplasm along the pipeline. So an example, so identifying a trait in say a glycine soja or a glycine tomentella, and that's about as far away from our elite germplasm genetically that you can get, and then making the crosses and moving that through the pipeline, that process of moving the identified trait into a commercially available variety may be 7 to 10 years. So that's a long-term deliverable.
That means that farmers get rather impatient in this process, I would think.
We can get impatient about it too when we're doing the research. And what that would mean too is you have students. Students are wonderful working hands, and they come in with brilliant ideas. Students and postdocs and academic professionals do a tremendous amount of the research that's reported at a university. A PhD student may be here for 6 years, may work on several projects, but never see that particular deliverable make its way into a variety. So it is something that goes through maybe a couple of different generations of students sometimes in getting to the farmer. So it's hard to be patient at both ends.
Well, we need to get to the portion where we get to actually walk around and see some of this in action. But I have a couple questions for you before we do that. And it strikes me, here you are, the daughter of an architect, and couldn't even begin to imagine that this is what you would be doing two or three decades later. But now you're an architect designing the future soybean, so to speak. I wanted to know what you think is the most significant or transformative change that you've seen in agriculture, and especially agricultural research, in these last couple decades.
Mm-hmm. Probably the most impactful change that's occurred in the soybean industry has been that in the area of soybean genomics.
what we spent quite a bit of time talking about here.
Yes, so understanding sequencing and knowing what and having information about genetic information, sequence information about the traits that are in the soybean genome has probably provided us with the most potential and future. Technologies like herbicide resistance, understanding how and why a soybean yields the way it does. You know, what are those genes? What do they do? We're getting close to that. So I think the most impactful event has been the sequencing of the soybean genome.
Okay.
Because that information can be used by many, many people in the public and private industry groups.
Okay, last question here is a different kind of question. Your kids grew up on the farm. How do you see the family farm evolving in the future? What do you see for the future of the farm in Illinois?
I think smaller farms are slowly disappearing, and the farms that are in existence are a little bit bigger. That would mean that there's fewer farmers and still the same amount of farmland. So the technologies that are available to farmers now allow them to farm more acres more efficiently than previously.
Is that a good thing, or do you have some nostalgia for the old-fashioned family farm?
I think if you're from the old-fashioned family farm, those new technologies can be a little heartbreaking. But when you see your children excited about GPS systems, that it kind of cuts both ways. Because I think, again, we were raised. My kids were raised on a farm. I wasn't raised on a farm, so we had a time there. I think to see some old things and farms the way that a farmer who'd lived his whole life grew up and came to understand it with the garden and the canning and smiling and having grit in your teeth and to see the new technologies would be a sad thing. But to see your children and the next generation so excited about new things on the horizon would offset the advances that a person might see as something.
You sound optimistic about the future of farming in Illinois, then.
Oh, yes. Oh, yes.
Well, let's take an opportunity now to cut the tape here, and we'll pick it up with you actually showing us some of the dynamic aspects that you've got in the laboratory and some of the other things going on here at the National Soybean Research Center.
Okay.
Thank you, Linda.
Thank you.